In this video, we will discuss about what are the strategies that can be used to treat the lysosomal storage diseases. And in the previous videos, we have discussed some of the lysosomal storage diseases. And in this video, we will discuss what are the strategies that can be used to treat those or to cure those diseases. So we will discuss the problem at every point in the during the central dogma of the cells, that is from transcription to translation of the genetic material into the final functional proteins. So the problem with the lysosomal storage disease is there is a gene mutation that has been occurred. In order to address this root cause, we can do the gene therapy and to correct the gene mutation that has been there in the lysosomal storage disease. So this is the first step where we can correct the problem. The next step is that if we are not able to correct the gene mutation, then this gene mutation might lead to a misfolded protein that might not be functional. So that thing can be improved by using the pharmacological chaperone information. As we know that chaperones are the proteins that are responsible or that helps the proteins to acquire their native structure, so that they will have a uniform post translation modification, and they will function as a normal protein. And this if this the problem is being addressed at this position, then this will take care of the degradation of mutant protein also, as degradation might occur when protein is not able to form their native structure, and the ERAD, that is endoplasmic reticulum associated degradation, will direct the non-native proteins towards lysosome, or towards proteasomal degradation. Next step is that this might lead to deficiency of lysosomal enzyme activities, because proteins are not in their native structure. So this might affect the enzyme activity of the lysosome, final lysosomal enzyme activity. And this thing can be addressed or can be compensated by increasing the lysosomal exocytosis. So instead of uh, accumulating the uh, lysosomal proteins within the lysosome, but if we can modify in such a way that lysosomal exocytosis increases, then this lysosomal storage content can be reduced, and all this will go to the extracellular matrix, where it can be easily taken care by other processes. And after these problems, the other problem that might acquire is that there is a intralysosomal storage of ungraded substrate, that is undesired, the substrates that are not mature enough to go towards the lysosomes. Because of the abnormalities, it might cross, and because of cross effects, it might leads to intralysosomal storage of ungraded substrate. And this thing can be addressed by inhibiting the substrate synthesis, that is substrate synthesis of such undesired or ungraded substrate can be inhibited using some strategies. And one thing that I left to tell you in previous disorders is that the lysosomal enzymes deficiencies can be taken care for some duration of time by enzyme replacement therapies. However, the problem in this enzyme replacement therapies is that if it has been done via transient transfection, then this need to be done after a certain period of time. This need to be repeat after a certain period of time. And so let's continue the other problem. Other problem is that because of the lysosomal disorders, it might uh, activates another cellular abnormalities. So these abnormalities need to be properly mapped, and their cross connection with the lysosomal storage disorders need to be identified so that corrective actions can be taken. One of the action that is mentioned here is to manipulate the autophagy. Autophagy is also one kind of degradation of the proteins. So whatever the abnormalities that are observed, that can be directed towards this autophagic pathway, so that can take care of this abnormal proteins and recycle them to the cellular machinery. And one strategy that has been great in that has been uh, that has been a constant topic of research among the uh, researchers is to transfer or is to transplant the donor stem cells directly to the patient so that have it can work at two it can work at two separate directions. First is that it will replace the it will try to replace the tissue that are abnormals within the patient, and another is that since the donor cells are healthy cells, they will secrete the lysosomal disease in the extracellular matrix, and these lysosomals, they will secrete the lysosomal enzymes, not disease. They will secrete the lysosomal enzymes in the extracellular matrix, and these enzymes will be taken by the abnormal cells of the patient, so that and so that this lysosomal enzyme can work in those abnormal cells also. However, these strategies are still under research and is yet to be established to a point that it can be used by a normal patient. So that's all for this video. I hope you got some information about lysosomal storage disease treatment and the various strategies that researchers are targeting. So that's all for this video. Thank you for your time.